Okay, all right. So hello, dear. So this is now the second part of your pre-recorded lecture in your classical chemical examination of urine. And for this part, we'll now start at the last the last test for protein, and that is your SBAX test or your quantitative test for protein. Now, again, recall ang ato na-discuss pa una are your qualitative, di ba? Your modified and heat and acetic acid and modified heat and acetic acid. And your semi-quantitative, which is your sulfosalicylic acid. And now we'll go now to the last type, which is quantitative. So when you say quantitative, we don't grade anymore. Like atong 3+, 4+, diretso na gyud siya ug concentration sa albumin na naa. So number na gyud, okay, concentration. So that is again your SBAX test. Now for SBAX test, we use a 24-hour filtered urine. Again, 24-hour para at least timed ang specimen. The concentration or tanang mga analytes, especially protein or albumin, which is what we're going to test here, kaya na-concentrate yun. So, mas ma-detect yun siya. Okay? So, 24-hour urine sample. And especially na quantitative siya, so it's really important na makuha nato ang total yun, ang total number or total concentration of albumin present. That is why we use a 24-hour urine sample. Okay. So, again, we filter it to remove other interferences, other substances, or other mga sediments man gani na naa sa urine sample. If qualitative test, di ba, usually we perform quantitative test ani if sa qualitative pa lang positive na gyud siya albumin or like positive siya albumin. Now if you want to determine the concentration of albumin gikan kag qualitative, you perform the quantitative, which is your SBAX. Now if you've performed already the qualitative test for albumin and then ang iyahang result is 3+3, we make a 1:5 dilution, and kung +2, we make tag 1:10 dilution. Because again, we want to make a dilution muna because again, too much manggod ang proteins na naa. Now, if ato siyang i-test dayon without making a dilution muna, it could be na dili tanang proteins ang mo react. I don't know if nakagets ra mo. Kay basin, too much siya kay ang protein against sa reagents na atong ibutang, right? So, if ever too much siya kay ang proteins na naa, we try to make a dilution first, okay? Para we know na dili gyud inana kadaghan ang protein na naa. And we'll make sure na at least mo react gyud siya sa reagents properly, okay? I do hope that's na kasabot ra na scenario, okay? So again, 1:5 dilution, 1:5, that's so 1 part urine to the total volume of 5. So pila ka distilled water? So 4 parts lang of distilled water. Okay? 4 parts. And of course, it's 1:10 dilution, 1 part urine, 9 parts na water. Okay, alright. So again, muna siya, dilution. Next, and of course, if the urine is not acidic, let's say sa reagent strip kay mo pH dili pa acidic or nag-pH meter baka. Okay? If di pa siya acidic, again, we add acetic acid. Because again, the purpose of adding acetic acid or the purpose of making the urine sample acidic is because, again, di ba, last video we mentioned that when proteins are exposed to acidic pH or heat, di ba, they tend to be denatured, or they are denatured. And once they are denatured, they precipitate out of the solution. So therefore, mas makita nato ilahang presence, right? Okay, so again, make it acidic. And then, we fill a tube, usually 15 ml tube atong gamiton. Okay, 15 ml. And then, ginalabel na siya daan na ay line for U, okay? U mark, and another sa taas is your R, okay? Alright. So, what you do is, of course, fill kag urine una, okay, until the U. And then another, after ni mag-fill og U, mo add ka og fill na po ka until the R, okay, your reagent, either SBAX or Chuchias. Okay? Alright. So, muna siya ang procedure. Again, urine sa una until the U mark, and then add a reagent until the R mark. Okay. Next, we cover the tube and invert 10 times. Invert slowly. Alright? And then, we place the tube vertically in a cool place and stand it for 24 hours. Okay? So, medyo dugay gyud siya. We stand it for 24 hours. And after 24 hours, we check for the precipitate. Okay, and there's a note there. We add pumice para mas dali magud ang sedimentation. When you say sedimentation, mas dali makombine ang mga ang ihi o ang reagent mismo, di ba? Ang reagent o ang ihi. So therefore, since increase the rate of sedimentation. So therefore, if na pumice, it will act as an accelerant. okay, sa pagmix sa reagent and sa protein, and therefore easy po ang pag-sediment, or meaning pag pagtapok sa ilalom, or para kanang easy ra pud siya mo precipitate. Okay, all right. So that's the purpose of your pumice. Okay, all right. Next, again, we measure now the height of the precipitate, and that is now equivalent to the amount of albumin in grams per liter. Now, I think, as far as I can remember, I think ang pag-measure is millimeters siya dagay to, or cm. I forgot. Okay, I'll have to clarify pa. But I think, yeah, pag-measure nimo, that's equivalent na again, that's equivalent na to the number or the concentration of albumin in your specimen, and that's in grams per L. Now, if you want to change it to grams percentage, you divide it by 10. Okay? Alright, and again, nag-dilute tayo, we multiply the final reading with the dilution factor. So if 1 is to 5, unsa may dilution factor? 5. And if 1 to 10, 10, di ba? Because again, we want to correct it this time. Because again, di ba, nag-dilute ta kay basin, taas siya kay concentration ng protein. So nag-dilute ta because we want to make sure na dili ra kayo daghan ang protein na ma-overwhelm ang reagents, di ba? Para mo react ra sila. Now this time, since nag-dilute matatong una, ato na po siyang i-correct karon. Okay, to account for the dilution na itong gigama. That's why we multiply it with your dilution factor. Okay, and of course, last, we dispose urine samples in our proper mga waste receptacles. Okay, and now for S-Box and Chuchillas, please take note ilahang composition. The S-Box reagent is composed of your picric, citric acid, and distilled water. And Chuchillas, phosphotungstic acid, 95% alcohol, and concentrated HCl. Because again, as you can see, ang common nila ay mga acids. Because again, with the notion that in acidic pH, proteins denature, and when they denature, they precipitate out of the solution. Okay? Alright. Ayan. So that's for S-Box. As you can see again, in a shorter procedure, pag-add nato sa S-Box, kung turbid ka siya, positive. Okay? Proteins, primarily albumin, and clear, which is negative. As you can see, ang reporting niya, muna siyang quantitative because it's expressed directly as a concentration. Dili na ka mag-grading na 3+, 4+. It's directly expressed as a concentration, either in grams per L, and if you want to change it to grams percentage, you divide it by 10. Okay, all right. And now we have a special type again of protein, as mentioned in the chemical exam, diba, na mentioned na nako. You have your Benz-Jones protein, the BJ. Buko juice. Sarap ng buko juice. Char. I miss the buko juice. Char. Okay, the Benz-Jones proteins or your BJ protein. Again, asa ni siya makitaan? It's seen in patients with multiple myeloma. And what is again multiple myeloma? Multiple myeloma is a type of cancer. It's an immunoproliferative. It's a proliferative, meaning kapadayon na yung divide ang cells. Yes, kapadayon na siya gama. Of unsa na cell? Plasma cells. And again, these plasma cells, they produce immunoglobulins, or inyong immunoglobulins known as your antibodies. Okay? And because of too much production of cells, the antibodies that they produce, or the immunoglobulins that they produce, are usually defective. Because again, daghan na kaayo, you know, inyong cytogenetics, di ba ka ng cell cycle, wala na proper regulation, wala na proper mga checking, whatever. So what happens is, ang ma-produce ng mga antibodies usually are defective or they're not functional. And in this case, your Benz-Jones proteins are a type of immunoglobulin, pero light chains lang. Because again, in your antibody, you will have this in your immunosero, second sem. Yes, one of my scariest subjects. Okay, anyway, your antibody, this is a structure of an antibody, a rough structure. You have your heavy chains, and you have the light chains. Okay? Alright. Bason, wala ko nasayop sa pag-label. Usually, this is your FC portion of the antibody, and this is the FAB. Okay? FAB. Ang FC portion, dira siya, muni ang mutapot sa antigen. Okay? And the FAB, usually, dira na mutapot ang other substances. But ang FC portion, dira dyan mutapot ang antigen. FC portion. Okay. Now, your light chains usually makitan sa FC, okay? And as you can see, since it's a monoclonal, meaning monoclonal, gikan ra siya usa ka type of cell, which is your plasma cell, monoclonal, right? And light chains ra siya, so therefore wala siya mga heavy chains, and ana ra siya na pagka-type of protein. So it's an incomplete na type of immunoglobulin, diba? All right. And they are seen again in multiple myeloma patients, or patients with multiple myeloma. Yes, so since, di ba, proliferative man siya, nag-produce silang daghan kaayo na inani, therefore, you know, renal threshold, it's too much na for the kidneys, di ba? Ang kidneys usahay mo, give up na po siya sa siging kadaghan na masakitan, daghan na levels of hurt, na ingnan mo ta na easy ang medtech? Charot lang. Okay, alright. So, again, kidneys, i-filter out na niya, appear na siya sa ihi. Okay? Because again, the tubular reabsorption capacity exceeds the levels of, or the levels of the protein or Benz Jones protein exceeds the tubular reabsorption capacity of your kidneys. Okay, and if for multiple myeloma patients, guys, ang imo jung gamiton is, or ang imo jung murag confirmatory test ana is your serum electrophoresis or immunoelectrophoresis. Now, I will not, dili na ako mo further anang electrophoresis. Because again, you'll have that in your ClinChem and sa IS soon. Medyo nalimot na bugo. But again, ang point lang sa electrophoresis kay you will have an anode and a cathode. So, mag-charge kang electricity. Imo nang i-charge yung electricity from here to there. And what happens is your fluid will move. Okay? Move siya from one place, from one end to another end. Then, ma-separate ang protein components, ana, depending on the different regions sa imuhang electrophoresis. Okay? Alright, so, again, in a nutshell lang to siya. Medyo, medyo very superficial. Wala kayo na in-depth na discussion. Okay? But again, that's for Benz Jones protein. That's a confirmatory test, but it can be seen again in urine, right? Usually for screening, it's not performed, pero it's easily detected by chemical methods. And again, unsa yung characteristics, Benz Jones proteins, they dissolve at 100 degrees Celsius, but they coagulate or they precipitate at 40 to 60 degrees Celsius. Okay, all right. So, here's an example. So, if ganahan ka na i-check ra gyud ang presence of Benz Jones protein, imong i-filter ang urine at 100 degrees Celsius, di ba? Kay ma-clear man siya, right? Ma-clear man siya. So, we filter it at 100 degrees Celsius. After filtering, ato siyang ipabalik or ipa-cool down ang specimen to 40 to 60 degrees Celsius. And if mo positive siya, then it's already your Benz Jones protein. Now, again, we start first with urine and acetate or your acetic acid ba? So we heat it at 56 degrees Celsius, di ba? Between ra siya sa 40 to 60. Of course, mo turbid siya because of the presence of protein. Alright? Now, we further heat it, di ba, protein. Kung turbid siya, negative siya because the cause of turbidity is your other proteins, di ba? Because if it's Benz Jones, dapat clear na siya at 100 degrees Celsius, right? Okay, so again, pwede mo na i-follow, and then pag abot diri, imo siyang i-filter. Alright? Imo siya i-filter para mawala ning other proteins, and then after filtering, you let it cool down, balik na po to 56 or 40 ba? And then if mo turbid gani, then positive na siya for Benz Jones protein. Alright? Okay, I hope that's clear. Alright, okay, and the last test, or the last chemical na itong ginatest, classically, sakto ba, is your glucose. Alright, so for glucose, again, reminder tamo, again, the renal threshold for glucose is 160 to 180 mg per dl. For glucose, again, for renal threshold, it's the blood level of an analyte at which the tubular reabsorption stops. Okay? And the substance now appears in urine. Okay? So if maabot ang level sa glucose at this or more than that, so mugawas na siya sa ihi. Okay? Because again, ni give up na, ni stop na. Dira na lang kutob si kidney sa pag-reabsorb sa glucose. Kaya it's too much. Okay? So if too much na gani, guys, let go. Ipagawas na sa ihi. Charot. Okay, alright. Please take note there, guys. In your ClinChem, you have conversion factors. So you have 0.0555. That's the conversion factor for glucose. So, asa nato siya i-convert? Mg per dl, 2 grams per liter. Tama ba? Okay, yeah. Basta, conversion factor of glucose is 0.0555. Okay, you will have that in your ClinChem. Daghan kayong conversion factors, guys. And usually, lumalabas sa boards. Okay, I think, ang nigawas sa amo ang boards na conversion factor is albumin, which is 10. Okay, mga proteins, mga total protein, mga globulins, albumin, 10 yun na siya, okay? Pero for glucose, it's 0.0555. Sa board exam, usually, mo ask sila, what is the conversion factor of glucose? Or iyahang ipa-convert yun, like magpa-solve sila. Muhatag sila, og example, result sa glucose na mg per dl, imong i-convert to example, grams per L. O, diba? So, imo na i-multiply og, dili grams per L, sorry. Millimol. Millimol per liter. Sorry. Tama ba? mg per dl. I'll just check. Nalimot ko. Basta, ang conversion factor niya, guys, is 0.0555. Okay. Alright, wala lang. Additional info. Okay. All right. So again, that's for the renal threshold of glucose. At this level or at more than that, mo pagawas na siya o glucose sa ihi. Because again, ni-give up na ang kidneys o reabsorb sa glucose because it's too much. Like, I can't handle this anymore. Bulag na ta. Char. Okay. Ipagawas na ikaw sa ihi. Alright, inana. Okay, so again, if too much na, ipagawas na sa ihi. Okay, charot lang. Alright, so again, for Benedict's test, we use, short ra siya. We use a tube again, 5 ml reagent, 5 ml of the Benedict's reagent, and then mo add tag 8 to 10 drops of urine sample, and we mix, i-shake lang siya gamay, and then i-boil siya vigorously for 2 minutes. And then allow it to cool, and we observe the result by observing any color change. Okay. And of course, dispose urine samples and perform necessary aftercare. Again, for Benedict's reagent, yung component, please take note, pinaka-important is copper sulfate. You have sodium citrate, anhydrous sodium carbonate, and distilled water. Okay, all right. So, unsa the principle ani? Now, the principle is glucose is a good example of a reducing sugar. So, by the name itself, reducing sugar, it reduces substances. Okay? All right. So, a very good example of reducing sugar is glucose. You have lactose, fructose, galactose, maltose. These are good examples of reducing sugars. Now, in the presence of, in a strongly alkaline solution, your reducing sugars or glucose reduces copper sulfate to copper hydroxide, which is yellow, or copper oxide, which is red. So, here's an example of reaction. The copper 2 ions found in your copper sulfate, reducing sugars in a hot alkaline solution, may mo siyang copper 1 ions. And mo combine siya with the OH or hydroxide to form copper hydroxide, which is a precipitate. Mo precipitate out of the solution, which is color yellow. Or furthermore pag yun, may mo siyang copper oxide, which is red. In the plus water. Okay? Basta ang point lang yun ana is i-reduce sa sugar, i-reduce sa glucose ang copper sulfate to copper hydroxide or copper oxide. Okay? Alright. Or cuprous oxide and cuprous hydroxide. Okay? Alright. Alright. So for reporting, as you can see, negative, clear, blue. Ang changes na is from blue to green to yellow and then finally mga orange to red precipitate. Of course, ang orange to red, inana nakadaghan ang glucose. Kaya daghan na ang ni-convert sa copper sulfate to copper oxide. Nahurot dyan niya convert ang copper sulfate to copper oxide or to cuprous oxide or cuprous hydroxide. Okay? And as you can see, muna approximate glucose concentration. So this is semi-quantitative. So ang pinakadaghan is greater than equal 2,000 mg per dl. Wow, grabe na yun. Okay? Alright, so here's an example of a picture. So you have negative, 1 plus, 2 plus, 2, 3 plus, and 4 plus. As you can see, muddy red na gyud siya na pagka-precipitate. Okay? Alright. Ayan. So that's for Benedict's. Very easy, very short lang gyud siya. Basta ang principle lang gyud niya is copper reduction. Okay? Your sugar, reducing sugars, glucose, reduces copper sulfate to cuprous hydroxide or cuprous oxide. Okay. And we also have what we call your tablet tests. Now, when do you say tablet tests? Tablet, as in like kanang ginainom nyo na tambal, pill, circle, tablet. And sa tablet mismo, nana siya yung mga substances there or mga reagents na naa. So it's very convenient, it's very easy. All right. So for each parameter, we have a specific tablet test. So for protein is Bumin test. For glucose, Clini test. Ketones, acid test. Don't forget acid test. Sumarasyon acetone. So ketones, acid test. And bilirubin, icto. Diba? Mga icteric samples. Icteric. Diba? Mga icto. Basta mga inanagani, it refers to bilirubin. So for bilirubin, icto test. Now for Clini test, guys, ang Clini test, it's composed of copper sulfate, sodium, sorry, wala na ako na-memorize, sodium carbonate. You have sodium hydroxide, and sodium citrate. Okay. Alright. Now, in the presence, imo na siya butang og tube. Okay? So, ang urine, butangan nimo og tube. Alright? And then, butangan nimo og, butangan nimo og ang urine, ibutang nimo ang urine sa tube. Alright? And then, ibutang nimo ang tablet inside the tube. Now, what happens is, in the presence of urine, and so, since naman siya water ang urine, no, mo-react ang sodium hydroxide and sodium citrate to produce heat. Okay? And this heat, diba, is needed, diba, sa ato ang, remember, sa ato ang principle sa copper reduction. clean test follows the principle of copper reduction. So, in the presence of heat, di ba, ma-convert niya ang copper sulfate to cuprous hydroxide and cuprous oxide. Okay, and sodium carbonate releases carbon dioxide para dili daw maka-interfere ang mga room air or other gases outside of the test tube sa test. Okay, all right. And this test is very exothermic, meaning mo release siya heat. So, init kayo ang test tube. So, therefore, dili mo siya dapat i-touch, okay, while it's happening. Now, we have what we call your pass-through, okay? Pass-through phenomenon, okay? Pass-through phenomenon, okay? So, this happens when taas kayo ang concentration sa glucose. What happens is, pagbutang mo sa tablet, instead na after mo color red ra siya for a while, di ba? Red or yellow or orange precipitate. And then after a while, mubalik siya to the negative na color, which is blue or green. That is known as your pass-through phenomenon. Because again, of too much glucose or too much sugar, wala nahurot dahil ng reagents, munang short ra kayo ang reaction, short ra ang precipitation sa red or katong orange ba na color. So nibalik dayon siya to the negative na color blue. So what you do is to prevent pass-through phenomenon, we add, instead of making 2 drops lang of urine, we add 5 drops of the urine. So instead of 2 drops ra, because again for clean test, 2 drops ra, we now make it to 5 drops. Okay? Para, of course, daghan ng glucose ang mo react sa reagent in a way. Para dili na siya kanang paspas lang reaction ba. Because again, ang sa 2 drops ang 2 drops, since gamay ra ang glucose, gamay ra ihang ma-convert na copper sulfate. So therefore, gamay ra pud ang precipitate na ma-create. Kay gamay raman ang glucose na available. Whereas kung 5 drops atong punan, so mas daghan na ang precipitate na ma-create, mas longer na ang precipitate reaction. Okay? Alright. Ayan. That's for clean test. Now, for acid test, ang acid test, guys, the components of your acid test is, of course, you have sodium, nitroprusside, di ba? Ato na po mga reagents sa sodium nitroprussides, reagents are put sa ato reagent strip. You have your glycine. Sorry naman, wala na akong memorize, no? You have disodium phosphate and lactate. Okay, of course, sodium nitroprusside, it reacts with your ketones. Glycine is added para ma-detect po ng acetone. And lactose is added para mas tsakto daw or mas makita ang color differentiation. Okay, and the acid test daw is very hygroscopic. So, if the tablet, ang buhato na mo sa acid test, kaya di mo siya ibutang og test tube. Like, mo siya ibutang og usa ka mat or like murag surface, and then mo drop ra ka og ihian ng tablet. Now, if within 30 seconds daw wala pa siya ni absorb, we need to change it because it's highly hygroscopic. Okay? So again, if within 30 seconds wala pa daw ni color reaction, ato siyang i-change because hygroscopic siya, meaning mo absorb siya dahil og water. Okay. Now, for glucose guys, or for clean test again, as mentioned, it measures reducing sugars. Now, reducing sugars are not only glucose. So therefore, pwede niya ma-detect ang galactose, lactose, fructose, and maltose. So dili kayo siya specific. A very good example, or asa nato pwedeng magamit ang clean test or unsa yang ma-detect aside from glucose is galactose. And galactose is if in the presence of urine, especially if pediatric patients, it means na siya inborn error of metabolism. Because we have an inborn error metabolism na walay enzyme na galactose 1 uridyl phosphotransferase, basta that enzyme, na mo break down sa galactose. Now, if dili na ma-detect sa newborn screening or sa pagkabata, pwedeng dili mo tubo ang bata because dili siya ma-digest ang galactose, or pwede po siyang mamatay because of improper digestion, improper metabolism. So, sa clean test, again, it's not only specific for glucose, pwede niyang ma-test ng other reducing sugars like galactose and glucose. And a significant ato is ang galactose gyud. Pwede po niya ma-detect ng lactose, pero makita daw na siya in patients or in babies na nag-lactate or nag-breastfeed, of course, because milk is composed of lactose na sugar, di ba? So, yeah. Another clinical significance lang sa clean test, aside from glucose, is galactose, because it could indicate an inborn error of metabolism. And last one is your icto test. Icto test, again, it's for bilirubin. You have, taas na kayo siya, guys. So, I'll just give you a separate handout for this para sa mga components sa tablets. Nganong ginamention ako ang components? Because in the board exam, usually, kanang if tulukaron ang mga board of examiners, muad sila ani. And then they will ask, okay, what are the composition? Or one of the following is not the composition of the acid test tablet, di ba? So, mga Morse code, di ba? So, at least kabalo na mo. Alright? Again, an ecto test, ang buhaton sa ecto test, guys, kay na ay mat, murag siya ka ng mat, murag ka ng mat. Okay, patunganan, and then ipatong nimo ang tablet here, and then what you do is, before nimo ipatong di ay, butangan sa nimo ihi, okay? 10 drops of urine, alright? And then after katong tablet. Now, ang positive reaction ana is blue to purple, okay? Blue to purple sa ilalom, okay? Alright, and if na mga colors outside of blue to purple, then it's considered to be negative. Alright, and if na mga interferences daw, pwede nimong butangan ni og distilled water para ma-wash out daw tong or ma-wash tong mga other interferences. Musulod siya sa mat because kaning mat mura siya ka na ganing kabantay mo anang basta mura siya na ay mura siya ka ng foam, mura siya foam, ina foam. So pag butan nimo og distilled water daw, katong ihi na imong gibutang diri, ang bilirubin na daw mabilin sa surface. Or ang mat itself, pag butang nimo sa ihi, bilirubin na daw ang mabilin sa surface, okay? But if napa daw interferences, pwede ka mo drop og distilled water para murag ma-wash out tong other interferences, ma-absorb siya sa mat, and then ang mabilin na lang is ang bilirubin sa surface. Okay, alright. So basically, that's it for your classical chemical exam. Again, we perform these tests to confirm. So, nag-reagent strip ka, kakita kag positive sa protein or glucose, and you want to confirm if nabagyod siya, then you can test this classical chemical exam. But again, this is not usually performed as I've experienced sa routine lab because again, it's time-consuming, kailangan kag daghang reagents and specimens, so musalig na gid sila. Usually, musalig na ta og reagent strips. As I mentioned, dili ra protein and glucose ang naay classical chemical exam. In your lecture ni Ma'am Bernal, daghan kaayo na like each parameter na siya mga tests gyud and specific reagents and the color reaction. So, prepare lang. Okay, watch out lang. Alright. But yeah, so ang ato na yung gidiscuss lang sa is, or ang ato na yung gidiscuss, and ato na po yung gina-perform gyud sa atong lab activity supposed to be is your protein ra gyud and glucose. Because again, they're quite, again, most usually common na mga significant or indicative of diseases in terms of chemical sa ato ang urine, di ba? Protein and glucose, and easy na po ng mga reagents. Pero again, as I mentioned, daghan gyud kayo, each parameter has their own classical chemical test. They have their own names, own reagents, and mga color reactions. So, watch out lagi mo sa lecture ni Ma'am B, ana, ginatabular na siya. Yes, okay. Alright, so basically, that's it for our lecture on this topic. I do hope clear ako mga explanations. Hopefully, bahalag na yung uban na medyo medyo lutaw, yes. Medyo gikan pa nangaway sa Twitter. Joke lang. Okay, all right. Again, yeah, so that's it for this topic. If you have any questions, again, feel free to chat in our group chat or PM me. All right, so I'll see you in our next pre-recorded lecture in your class. So have a great day, guys, and keep safe.